Well, it's 12 o'clock, but if you're having lunch right now, you can't bring it into the Schroeder Lab. Welcome back. We're up on the fourth floor now at BTI, meeting with members of the Schroeder Lab, who is doing some really interesting research. That word interesting is getting thrown around a lot today. It's probably losing its appeal, but it's all interesting here at BTI, and that's why we're bringing you these live streams. So I'm here with Josh from the Schroeder Lab, and he's going to take us around and tell us all about this innovative work with roundworms and other things that they're working on here. So Josh, tell us a little bit about kind of like an overview of what the Schroeder Lab is all about.
Okay, so the overview of the Schroeder Lab would be that we are interested in the chemistry of life. So we want to understand how organisms use chemistry to function as an individual and to function as part of the community more ecologically. So the main focus of the lab is really working with the model organism C. elegans, the nematode, and now other nematodes as well have kind of moved into the focus. And what Frank and others have done here is sort of discover signaling molecules that play all sorts of roles in their behavior that were previously unknown. And so that can have a lot of important implications for humans, since we share some similar biochemical pathways. Now,
yeah, I'm sorry, I don't mean to interrupt you, but I think that's one of the things I find most fascinating about this lab is maybe some people that aren't deep in the science, they might be surprised to hear that there could be so many similarities between the chemistry of a 1 millimeter worm and humans.
Right, yes, that is always surprising. But it just turns out to be true that things were conserved for quite a distance on the evolutionary tree. Myself, actually, I don't work with C. elegans, so that's sort of the other part of the lab. There's a smaller part which consists of me and a couple other people, and we're more interested in studying the chemistry. So in particular, we work with Aspergillus fumigatus, which is the most notable human pathogenic fungus. And in the same sense, just analogously to discovering signaling molecules in C. elegans, in Aspergillus, we're interested in how this organism uses chemistry to sort of help itself as a pathogen. So if we have a sickness and you go to the doctor, they give you a drug and they say, this is a molecule that will disrupt some part of that pathogen's ability to survive. Now, conversely, when an Aspergillus infection occurs, it actually makes its own drugs, essentially, to stop your immune system from defending yourself. And so for people who are immunocompromised, this turns out to be extremely bad. They're very successful. They have the ability to continue to grow inside the human's lungs and eventually spread around and most times kill the person. So what we've been doing here is sort of looking at the biochemical pathways that are involved in the fungus's ability to make its own drugs, I'll say, to help its survival. And so just coincidentally, by doing that, we not only discover sort of interesting ways of how it is helping itself survive in a virulent setting, but we also discover other chemistry that can actually help us as chemists ourselves. So probably a lot of what you've heard about today has been related to plants genetics. And what most people know is that plants produce a lot of isoquinoline molecules. So some of the drugs like morphine, codeine, they come from a pathway in plants that's very well studied and established for many years. And we've taken advantage of that to produce all these painkillers. And what we found in my work specifically is that Aspergillus and many other fungi actually make similar compounds with similar biochemical steps, which was very surprising and very interesting. And it represents a sort of convergent evolution where you have two different kingdoms of life that have evolved separately to make the same sort of class of molecules. And so these are probably important. And we don't understand what their role is yet in the fungus, but it's something we hope to do. So I actually just graduated, so I'm moving on in a month to do a postdoc. So I can show you around the lab a little bit.
Where are you off to?
So I'm going to Caltech.
Okay.
So I'm switching fields. I'm going to basically work with stem cells and do some more biological stuff and some materials work as well.
Awesome. Well,
So
congrats on that.
Yeah, so that's kind of the gist of that's what I do, and then the nematode side, and I can sort of, there's one other side of this lab that I haven't discussed yet that we can talk about inside here. So sort of some of the important pieces of the lab here, you have incubators, so these regularly use, so we just grow large cultures of worms. So people don't expect it. That's how things are actually growing.
The worms don't get dizzy.
The worms don't get dizzy. They have to move. If they're not moving, they're not. We will often culture. This is an example right here in this glass. And those get fed to the worms in cultures like this. And then the worms grow, and then we process them and use analytical chemistry tools to discover the type of molecules they're making under certain circumstances. And most everything we do is always coming back to a genetic context. So in my work in particular, everything I do is in collaboration with fungal geneticists at Madison, Wisconsin. And they sort of do the genetics to make a mutation, and then I can look and see how that affects the molecules that the fungus is making. So it's important we always have to grow things. If we move down this way, yes, you probably eat them all the time. We're all consuming them regularly. And in fact, probably if you're not going to be as healthy. So mostly, from most of the evidence we have is that a healthy worm diet makes a healthy person. So we can come around to the other side and maybe introduce another member of the lab. So this is Nabi. He's our mass spec specialist. So I think we'll go in there momentarily. Maybe you can have something to say over there. Over here, we have our hoods. So the last part of the lab that I didn't really get a chance to talk about a moment ago is that not only do we discover the molecules here, but we also want to make lots of them. So some part of our lab is dedicated to synthesizing small molecules that are signaling molecules in worms or in fungi. And of course, most of all that work happens inside a hood, as much as needs to for safety. And I do a little bit myself, but not that much. But someone who's really their focus is on making these molecules is another member, Kitty, who's right in the middle of doing something, but she can talk. So Kitty, tell them a little bit about how you're making signaling molecules for C. elegans and other nematodes. Tell them about what you do.
Okay, so we want to study the bioactivity of the ascarotus because most of them are related to aging, and understand them will help us understand ourselves, not only the small worms we study. So I want to make them with the best efficiency and as high as possible yield, so that we can use the pure chemicals to study their bioactivities in vitro. That's what I do.
And what does that look like in
Um,
terms of, is that something you're doing here on this space?
so what I'm doing now, I just finished running a column. I'm trying to purify the compound I just made 2 days ago, because we want really pure samples for the bioassay we're gonna do. So that's what I'm doing now. Those are different fractions. So the machines you just pass by are the copy flash. That's what I use like almost every day. Yeah.
So I can tell you a little more about them.
Thank you.
Thanks, Kitty. So as Kitty just said, one of the most important pieces of equipment that we have in our lab is a host of so-called combi flashes. And these are automated normal and reverse phase chromatography for large-scale separations of mixtures. So the synthetic people typically use what we call the normal phase, and then someone like myself would use what we call the reverse phase. And that's just related to the types of molecules that you want to separate. So they're optimized for a certain polarity range depending on what you're interested in doing. So this is a big, it's very important to us because any of you out there doing chemistry or maybe thinking about doing chemistry, you may have learned to do chromatography manually, where you would have to take a glass version of these two things, pack them with silica yourself, and actually pour in solvents and then run a column with your hands underneath it. I actually did that for a couple years as an undergraduate before I came here. And these things are lifesavers, as anybody will know. You can load your samples, set it up, walk away, come back, everything's separated. Doesn't work perfectly every time, but for the most part, it's amazing. So that's an important part of our lab for everyone. And then in here we have a newer piece of equipment. So we have more incubators. These are the fungal incubators, so these are the ones that I mostly use. We don't tend to mix the incubators because the fungus, they make millions and millions of tiny, tiny spores, and that's ultimately what they grow from. And so those spores are airborne, and they are everywhere. They're here right now in this room. We're breathing them in, and our lungs are dealing with them. Our immune system is essentially engulfing them and killing them. So if we were to mix the worms with incubators where there was fungus, most of the time the fungus ends up in the worm cultures and pretty much kills everything. They're the best growers. They usually dominate the cultures. So then here is a new piece of equipment, brand new, thanks to all the great funding from the NIH and HHMI. We have the ability to buy not only combi flashes, but other important pieces of equipment. This is what's called a speed vac, supplied by Thermo. So we just got this like 2 days ago. We're super excited about it because in the course of our research, we have to dry samples. So we have samples that are in solvent, in vials or flasks, and we have to dry them. And what we used to use and still will use to some extent is another piece of equipment that's right behind you, Keith. So this, many people may be familiar with the rotovap. So if we go over here, basically what this does is rotates the sample, and then a vacuum pump pulls vacuum. It pulls off the solvent that gets condensed here, collected in the trap, and discarded. So you can do one sample at a time. In my PhD, I probably rotovapped several thousand samples, one at a time. Now that I'm leaving, we have this, which can hold 100 samples at once and spin them and dry them. So you can imagine that's going to increase the efficiency for a lot of people.
So you're saying you could have used this a little sooner?
Oh yeah, I could have, but it's okay. We got it done. So now we go to another part of our lab, although it's a shared space in BTI. This is the mass spec facility, which was not officially on the tour, but the Schroeder Lab more or less takes responsibility for it, since we were the origin and sort of the main motivation for getting this piece of equipment, along with others like the Jander Lab, mostly. So basically what we have here is a couple of different related instruments, we have a regular LCMS with a very old mass spectrometer that's very reliable. That's about 20 years old. It still works, and it's been working well, and I've maintained it my whole time here. So what we do there is the same thing that you do with the large-scale combi flashes that I just told you about. However, on a much smaller scale with a much higher level of precision, and in conjunction to the separation of the molecules, we're actually analyzing them to determine what their molecular formula is, to determine how much they weigh. Their mass to charge ratio is measured there. And so we have two of those that are very similar, one over there, and these are kind of in the $50,000 range of pieces of equipment. So they're older, but they're just workhorses. They're basically running all day, every day. And then more recently, we've really amped up the facility with the addition of this instrument here, which is the Orbitrap. So this is a Q Exactive from Thermo, hooked up to a Thermo Ultimate, originally made by Dionix, UHPLC. So this is a really high quality mass spectrometer that allows us to get very precise mass measurements for all sorts of molecules. And this is really sort of changing the pace at which we do our research, because our ultimate goal is to sort of comprehensively understand what all the molecules are that C. elegans can make, that fungus can make, and even maybe that we can make. Eventually, hopefully, we'll get there. And this instrument has basically sped that up maybe tenfold. And we're getting so much data where we actually have to learn how to deal with the data now. So going forward, some of the most important things are going to be handling all the data that comes from these types of instruments. And people all over are getting into these and really taking advantage. So we appreciate that very much. BTI helped us get this. It was a large group effort spearheaded by Frank, my advisor. I think everybody here has benefited from it. Most of the labs have started to use it. And this too runs 24/7, 365. So we're busy with that. Okay.
So that seems to be a really good example of people sometimes maybe think of, well, how does the evolution of technology help science? That seems like a pretty good example.
Yes, that is a great example. And another great example, which we can't show you, which is also a workhorse of our lab, would be the NMR at Syracuse and over in the chemistry department. So we use a lot of NMR, and those are just large. Basically, it's an MRI for not for people, but for very small samples. That's the simplest way to explain it. And we don't have one here, but that's another example where recent spectrometers, especially one that we have really enabled us to solve structures for amounts of sample that we couldn't do before. And so we're making a lot more progress because of that. So we thank the physics community thoroughly here, because most all those things come originally from physicists. Okay, so let's move this way to the last part of this tour. And cover this. Okay, well, there's nobody here.
What are the odds?
Where is everybody in the Schroeder Lab today?
What are the odds that there's a camera around, everyone goes clean?
Yeah, so this is not my domain, but it is part of the Schroeder Lab. This is sort of the part of the lab that does all of the molecular biology. So some of the other people you heard speak today are mostly biologists, plant biologists. They do a lot of molecular biology where they're working with RNA, DNA, or proteins, or biochemistry. So, of course, a significant portion of our lab specializes in sort of doing those types of things, cloning, making mutations, expressing proteins in bacteria from C. elegans or from fungi, and sort of studying them in more depth. So a lot of the stuff they use is in this room. It obviously looks much more simple, and everything's sort of smaller than for the chemists, but the bills are just as high. Everything's very expensive for their reagents.
Yeah.
Okay, well, I wish somebody else was here to talk more about it, but that's kind of...
Well, we appreciate it, Josh, all the thoroughness of the tour, even though some of it might have been out of your comfort zone. Did an amazing job. And you can certainly learn more about the Schroeder Lab work if you go to our website, bti.cornell.edu. Look under faculty or under research. You'll see Frank Schroeder, and you'll see lab members like Josh and all the important, really impressive work that they're doing here with regards to the chemistry of life. And it's interesting that all of this technology and all of these, all this machinery all kind of boils down to that. So it's it may look technological, but it's very kind of organic at the same time, which I think is pretty interesting. We're going to take a whole half-hour break before our next live stream, so come on back then. I also wanted to announce that we have a match program going on right now until 3 o'clock. You'll see more about that on our Facebook page, but for people that support our campaign between now and 3 o'clock, if we can raise $300 in the next 2.5 hours, our board member, Paul Comet, is going to match $300 in support of mentorship here at BTI. So if you're thinking about getting in on the Giving Tuesday, now would be an excellent time. All right, we'll see you back here in a half hour.
Thanks.